before beginning my presentation, I should acknowledge the original creators of the content which I used for this presentation. And this presentation is solely for the purpose of knowledge sharing and educational purpose. So as the introduction was given, that the talk will be on like fixing the tangled thread around the untargeted and or targeted metabolomics. So how do we start it? I started with the story. What kind of story is this? This story is all about the metabolome. How it started? It started in 1998 when the term metabolome was coined, and then the discoveries had happened because of the technological development which went hand in hand. So what kind of technologies were there or are there? So you can see that the number of publications across these last 15 years rose from few hundred to few thousands in the field of metabolomics. So the GC and the NMR, these were the techniques, or these are the techniques which really contributed a lot in the field of metabolomics. I'll deal with mass spectrometry separately in the subsequent slides, but there are some problems with these technologies. What kind of problem these technologies are facing? If I talk about the gas chromatography, then we can handle only volatile compounds. If at all, if we are able to derivatize, then derivatization itself is a lengthy process. Then there is a problem with the thermolabile compounds. So if we talk about metabolome, then most of the molecules, or essentially all of the molecules, are endogenous in nature and they may be thermolabile in nature. So then what is the problem with NMR? The NMR has low sensitivity, poor resolution, and the instrument is extremely costly. So what could be the game changer? The game changer here is the mass spectrometry. So if I talk about mass spectrometry, then I should give you some details about the current mass spectrometer which is there in my laboratory, maybe in your many of your laboratories. And the mass spectrometer which you are seeing today is sitting with 6 Nobels. And I should not say that only these Nobel discoveries are at the mass spectrometer which you are currently seeing. Yes, there are remarkable contributions from other inventors which really paved the way. So these 6 Nobels. At the left side, if I start from J.J. Thomson, to the right side, Koichi Tanaka. So what J.J. Thomson did? There is a small video. So let me start it. So in this video, you can see that this gentleman is trying to replicate what J.J. Thomson tried to do. A cathode ray is there and he is applying the magnet, and because of the application of this magnet, there is a deflection in the cathode ray. The similar kind of experiment was performed by J.J. Thomson. And this is a replica of the gas discharge tube which J.J. Thomson used for the discovery of electron, for which he got the Nobel. And the same kind of cathode ray you must have seen in the old television when you are popping from the back, then you can see that arc lamp, sort of like red color of lamp, is glowing over there. So this is the magnified image of that. From here, the another invention, remarkable invention, Austin. He took the lead from the J.J. Thomson, and then he made first mass spectrometer. Then I'm going in chronological order. So the Demelt and Paul, they both got the Nobel for their discovery of trap. So how this trap works? So here is a animation through which you can easily understand how trap works. And the mass spectrometer which I have is 6500 plus Q trap. So it has trap function, and I know that when you apply this trap, then what kind of differences you see. So if a molecule is going, then you get a signal. The other molecule, it enters and you get the signal. But when you activate the trap, then actually what is happening? The molecules are getting trapped into the, are they are being held for a certain period of time. So you are like accumulating the molecule and then you are releasing at one go to the detector and your signal is getting enhanced. And I must tell you that in many of the cases, this enhanced mode, because of this activation of trap, really help us in identifying lot many things. Now, I should move on to the next discovery by John B. Fenn. And the discovery of John B. Fenn actually uh, like put him into trouble, and he had a like litigation with the university where he was working. But the discovery which he made, the electrospray ionization, really changed the mass spectrometry field. And because of this discovery, the liquid chromatography came into picture, and now most people use liquid chromatography in association or in tandem with mass spectrometry. So how this liquid chromatography actually works, or this ESI actually works. So you are introducing your liquid sample through this small needle, and this needle is kept at very high voltage, either in a positive mode or in negative mode. And when your molecule, analyte of interest, are traveling from this small tube, then they are getting ionized because you have kept it at very high voltage. And once they get ionized, like the clusters are there and the solvent molecules are also there. Now they are traveling, and the heated gas is also passing through. So because of that effect of that heated gas, your solvent molecules are getting evaporated. So desolvation is happening. And at a certain point, there will be a Coulombic explosion because the like-like molecule or the like-like charge are coming quite near to each other. So Coulombic explosion will be there, and then you have a generation of multivalent ions. Then finally, those ions will enter inside the mass spectrometer. So here, if I tell you that how it is different from the gas chromatography, there you are directly converting that into gas, but here it's indirect drying. So you are like you are like you are preventing yourself, or you are preventing your molecules from thermolabile molecules from degradation, because you can increase or decrease the temperature on the basis of your analyte of interest. And when you see from the source window, you could see that some sort of like this kind of discharge you could see. And the second one, so the second discovery was made by the Koichi Tanaka. Koichi Tanaka was working on macromolecules, the work which was given by his company Shimadzu to him, that you need to work upon macromolecule and you need to figure out that how we can identify macromolecules. And he wanted to, like, he was testing lot many matrices, and then he invented one, and for that one he got the Nobel. But that paved the way for him to the molding which we are seeing nowadays. So from here, let me talk something about the resolution, because it is all about resolution when we talk about the untargeted and the other side targeted. Though what kind of difference this untargeted and targeted words are making. So here at the left side, you can see there are two masses which are quite near to each other, but there is a difference of like 1 Dalton between them. So when you go on to calculate the resolution, then you get the resolution somewhere around 1000. And you see these masses, which are like different from each other at the decimal level, after the decimal level. So when I calculate the resolution power of these differences by using the same formulae, I get the resolution of somewhere around 8000. So here, what kind of difference we are trying to make? So here, mass spectrometry can be divided into two like branches, or two distinctive instruments. One is high resolution type of instrument, which will follow this one, and another side, the low resolution type of instrument but high precision type of instrument, which will be following this one. And let me ask you one question, whether the resolution is the answer. Maybe in some cases, but the moment you are increasing your resolution, you have the fear of losing quantification power. I'll explain you in much detail in later slides. From here, let's move on to that how this unit mass resolution, or I would say that this low resolution mass spectrometer or high precision mass spectrometer looks from inside. So this is the instrument which is there in my laboratory, and it is actually the series arrangement of quadrupoles. So quadrupoles are what? They are 4 rods you can see over here, which are like alternatively having the alternative polarity. And what this alternative polarity actually does, it gives the molecule, it gives the ion a circular motion. And because of this circular motion, in quadrupole 1, you can select your parent ion. Then in the quadrupole 2, you will be having the fragmentation generation. Your daughter ion or your fragment ion will be generated. And in quadrupole 3, you will have again the selection. And by that way, you can select your parent here, then your fragment here, then it will be detected. And I must tell you that the quadrupole has the unit mass resolution. And because of this, the instrument is known as low resolution or high precision mass spec. Because why it is high precision? Because you are locking your mass here, you are locking your mass, daughter mass here. And by that way, you get high precision. And the instrument from inside looks like this. Now let's move on to the next instrument. And here again, on the basis of resolution power, the instrument can be divided into like 2 parts, I would say. So first of all, let's see this video. So it is all about the Orbitrap Exploris 120. It is the instrument which is based upon the Orbitrap type of technology. And as I talk to you regarding the ASI electrospray ionization, which you can see over here, this electrospray ionization is generating the ions, but there could be some particles which may not be charged. So because of this S-shaped structure, the molecules which are not charged, they will be like gone over here. And now this is the heart of the instrument, the orbit trap, where the molecules are, the charged ions will be entering, and they will be rotated at very high speed in this circular motion, and there will be a separation in horizontal as well as in vertical axis, and you will get like very good resolution power. And the another instrument, which you are seeing here, it is the time of flight.
The multi-tough process is a 2-phase procedure. Initially, the samples are fixed in a crystalline matrix. In the initial ionization step, they're bombarded by a laser. The sample molecules vaporize into the vacuum while being ionized at the same time. High voltage is then applied to accelerate the charged particles. The second step is the time of flight mass spectrometry phase. In the linear mode, particles will impinge upon the linear detector within a few nanoseconds after ionization. Higher mass molecules will arrive later than lighter ones. Flight time measurements make it possible to determine molecular masses directly. Each peak in the spectrum corresponds to the specific mass of the particle.
So to put it in a nutshell, if you are allowing a molecule to run for a marathon, then in between the molecular weight difference, you'll be able to get a quite well difference in between the molecule which are having quite near molecular weight. And so there is a this is the comparison slide between the high resolution mass spec at the left side and low resolution mass spec at the right side. So you get this kind of data, and most of my previous speakers have shown this kind of data, the HRMS attained data. And in HRMS, what did you get? what do you get? In HRMS, you get high coverage because it is always focusing on lot many molecules. It is trying to collect as much of data as possible. And because of this collection, what it loses the repeatability. When you are repeating the samples, then you may find the difference between the abundance or in between the the counts which you are getting, and the linear range. Also, there is a limit between of the linear range, but there are instruments which are coming with quite a good linear range, but there is no match with the LRMS type of instrument, where it has the wide linear range, good repeatability, but the coverage is poor because, as I told you, that they are coming with the unit mass resolution because of the quadrupole arrangement. So now let me give you some insight into the metabolome. So you have quite a good idea about metabolomes, that it starts from the genome, then transcriptome, proteome, and then finally metabolome. So it actually gives you the phenotypic information that what role environment is playing in how your body is like reacting to whatever environment changes are there, or environment effects are there. So if I tell you that what is the ancient example about the metabolomics, then that comes from my country, India, where the physician used to tell the patient to collect their urine and to put it in open. So if the ants are getting attracted towards the urine, then they say that yes, you may be having diabetes because the sugar is getting secreted in the urine and the ants are attracted towards it. So it's a kind of sort of targeted type of metabolomics which they could see. And another example is, if in case of gout, where the uric acid accumulation is there in the body, so you are pinpointing to one metabolite. If I take another example, then creatinine is a good example of your kidney function test.
Keeping it's 2 more minutes, please.
Sure. So this is the example which I'm taking from my own lab, where we were working on the ADA2 deficiency patients, and the ADA2 deficiency is actually an inherited autoimmune sort of disease where vasculopathy happens. And when we were working upon this disease, then we wanted to see how inosine levels are different between two phenotypes. So first we made a targeted method for inosine quantification. And when we saw between the patient, the control subject, and the ADA2 deficient subject, then we could see a quite good difference between the inosine level. Surprisingly, in one of the patient, the genetic report said that no, ADA2 deficiency is not there, the genetic alteration is not there. But when we subjected, because of the the diagnosis or because of the symptoms which the patient had, so we subjected for the same test, and we could see that yes, the quantitatively inosine was low, and even in the patient, the parents also, the inosine levels were low. And the another example is of untargeted type of analysis, in which we recruited rheumatic heart disease patients, and we got results, and the pathways which were dysregulated in this case were also there. And here, very interesting catch is there. Although the data is quite old, but still it has the value. In E. coli, human and plants, these are the metabolites which are there. But how many metabolites we are able to identify till now are quite a few. So this pathway analysis, when I go into it, or when I enter into it, then it looks very clumsy. It looks very difficult. But you can simplify this by putting some major pathways like fatty acid synthesis, pentose pathway, so on and so forth. And I must tell you that the lipids are also the part of metabolome. When you do the metabolome, then lipids also come into the metabolome only. And how biocrates is helping, because I also worked upon lot many products of biocrates, although I did not show the data because they are still into like a publication. So they are quantitative as well as qualitative, but the beauty with the quantitative kits are there that you get the data, you get the quantity at the end of the day. You can use it for population studies and for other studies as well. So I always say that whatever we have detected so far is only the tip of iceberg, and me, you, and all people who are working in the field of metabolomics or multiomics will be adding more data to it. Here, a very important, although it is not related to this presentation, but I want to show this presentation. I want to show this video. So by educating people, we can save, if we are able to save one life, then that is more than enough for us. So my last slide, metabolomics and lipidomics, they are actually giving you the phenotypical information. The choice between untargeted and targeted is actually the deciding factor will be the cost and the what kind of outcome you want to have. And if you have any, if you want to contact me, then you can contact me and you can mail me at my email ID. Thank you.